Program Bachelor of Science, first year, subject Botany, semester 2, course code BOC 102, course title Biodiversity 2, Vascular Plants. Title of the unit Gymnosperm, module name Reproduction of Pinus 2. Myself Sushma Salgaonkar, MSc, Assistant Professor in Ganpat Parsikar College of Education, Harmal, Goa. Outline: Introduction, gametophytic phase of reproduction, male gametophyte and its structure, female gametophyte and its structure, learning outcomes. In this, student will able to describe gametophytic phase of reproduction, explains male gametophyte and its structure, explains female gametophyte and its structure, and also able to describe life cycle of Pinus plant. Reproduction. Reproduction of Pinus is divided into sporophytic phase and gametophytic phase. In both these phases complete the life cycle of Pinus, wherein in this module we are going to study about gametophytic phase of Pinus. Introduction. Pinus is heterosporous, which produces two types of spore. One is microspore, pollen grain. Second is megaspore, embryo sac. Both microspore and megaspore is the last stage of sporophytic generation and first phase of gametophytic generation. Strobili. There are two types of strobili, male and female. Male will develop microspore and female cone will develop megaspore. This is the sporophytic phase of the Pinus plant. Microspore will develop male gametophyte and megaspore will develop female gametophyte, which is still going to retain on the megasporangia, which will lead to the formation of two or more archegonia. So this is the gametophytic phase of the Pinus plant. Male gametophyte. The microspore, pollen grain, is the basic unit of the male gametophyte. It is an uninucleated structure with two asymmetrical placed air bladders. The air bladder is also known as wings or saccus and arises on the lateral side of the microspore. Each pollen grain has circularized outer layer called as exine and inner thin layer called as intine. The outer wall of the wings has a reticulate sculpture. Male gametophyte develop partially in the microsporangium and partially in the ovule. Mature microspore. Mature pollen grain is composed of two ear-like sac on the outer side, a large tube cell, a small generative cell, and two tiny prothelial cells. This is the origin of microspore, wherein LS of male cone is given, which will give rise to microsporophyll, and inside the microsporophyll, wherein your microsporangia is present, in that your microspore are going to develop. This is the photograph of mature male cone, the section of megasporophyll and microspore. Development of female gametophyte. The microspore develop in the microsporangium. Within the microsporangium, microsporocytes cells divide by meiosis to produce 4 haploid microspore. Further, mitosis of this microspore produces 2 nuclei, that is the generative cell and the tube nucleus, which will develop into male gametophyte. Upon maturity, the male gametophyte is released from the male cone and it reaches the ovule due to the pollination for the further development. A number of microspore reaches the ovule where they are attached with the sticky mucilage substance from the micropyle. When the mucilage substance dries up, some of the microspores are drawn inside on the apex of the nucleus. Later on, the spore coat splits between the wings and then the tube cell protrudes and grows from the pollen tube. Ultimately, the pollen tube penetrates the nucleus. The generative cell then divides to form a sterile stalk cell and a fertile body cell, that is spermatogenous cell. So the body cell further divides to form non-motile unequal male gametes or sperms. Female gametophyte. The haploid megaspore is the first cell of female gametophyte. It is never shed and retained permanently within the nucellus. Archegonia is present in the female gametophyte. Development of female gametophyte. In the female cone, megaspore mother cells are present in megasporangium. The MMC, that is megaspore mother cell, divide by meiosis to produce 4 haploid megaspore. One of the functional megaspore divide to form the multicellular female gametophyte, while the rest are degenerated. Origin of megaspore. This is the LS of female cone which will give rise to megasporophyll, in which your megaspore is going to develop. This is the cross section of female cone, then single megasporophyll, and inside that megasporophyll mother cells are present. Female gametophyte stages, which is divided into nucellar stage, wherein megaspore undergo 11 successive free nuclear division and produces nearly 2000 small nuclei. And second stage is cellular stage, wherein the nuclei comes to lie towards the periphery due to the enlargement of the central vacuole, which later disappears. After pollination, the development of the female gametophyte takes place again. The walls are broken down around the nuclei and ultimately a solid mass of thin walled cells are formed. The massive tissue thus formed within the megaspore, which is called as female gametophyte. It is often referred to as the endosperm, which is haploid in nature. A few flask-shaped archegonia, 2 to 3 cells are formed from the superficial cell, that is archegonial initials, that lie toward the micropyle end of the female gametophyte. A mature archegonium consists of a neck of 8 cells, 1 ventral canal cell and 1 large egg. Fertilization. After a year of pollination, fertilization occurs. The pollen tube moves downwardly and reaches the neck of the archegonium. It then penetrates the neck and the tip of which bursts to discharge the 2 male gametes. One of the male gamete unites with the egg to form diploid zygote. Seed formation. It is formed from the mature ovule. Winged seed escaped from the cone are blown away by the wind. Example, seeds of Pinus girardiana, commonly known as chilgoza, used as a human food. Seed germination is apigeal, which means cotyledon comes above the ground by the elongation of hypocotyle. Life cycle of Pinus. Life cycle of Pinus show alteration of generation. The sporophytic phase is of long duration and represented as a huge pine tree which bears staminate and ovulate cone. The life cycle of Pinus. In this, branch of mature Pinus plant shows female cone, which is made up of megasporophyll. On the megasporophyll, megasporangia is present with an ovule. This megaspore mother cell, which is haploid, is developed inside that megasporangia. On the same branch, male cone is present, which is made up of microsporophyll. Within the microsporophyll, microsporangia is present, and within that microsporangia, microspore mother cells are developed. Both will undergo reduction division, that is meiosis. First one, functional megaspore, will give rise to free nuclear stages and then cellular stages to form female gametophyte, which is mature ovule. Within that ovule, archegonium is present, and within the archegonia, egg is developed. Microspore. Tetrad of microspores are formed, which will lead to the formation of mature microspore, which will develop into pollen tube. Within the pollen tube, sperm cells are generated. These sperm cells and egg cell will fertilize together to develop zygote. This zygote will develop into further stages to lead to the formation of embryo, and this embryo will give rise to germinating seed. And finally, we are going to get the sporophytic plant body. So, the whole sporophytic generation which is diploid in nature, and gametophytic generation, which is haploid in nature. So the life cycle of the Pinus, which is given in the summary: a Pinus tree, the mature sporophyte is diploid. It will produce both seed cone and pollen cones because it is monoecious. Seed cones will produce 2 megasporangia on each oviviparous scale, each one surrounded by an integument. Within the megasporangia, a megaspore mother cell undergo meiosis to produce 4 megaspore, which is haploid. 3 die and 1 remains, developing by mitosis into the megametophyte. The megametophyte produces 2 archegonia, each with an egg. The pollen cone microsporangia are produced on each microsporophyll of the pollen cone. Initially, they are filled with microspore mother cell, which is diploid, and then undergo meiosis to produce microspore, which is haploid. These microspore grow by mitosis, though only 2 round, into 4-celled microgametophyte, that is pollen grain. Each pollen grain is composed of 2 ear-like air sac on the outer side and a large tube, a small generative cell, and 2 tiny prothelial cells. The mature pollen grains are dispersed on the wind to the seed cone, where the tube cell will form a pollen tube into the megametophyte by entering through a gap in the integument called micropyle. The generative cell divide to produce the sperm cells, which travel down the pollen tube to fertilize an egg. Once fertilized, the integument closes, forming the seed coat. The embryo develops within the seed, consuming the megametophyte and the megasporangium, now called as the nucellus, as it grows. The seed is dispersed, and if the right conditions are present, then the embryo emerges from the seed coat and develops into mature sporophyte, that is your plant body. References. Thank you.